Hi, good morning, Year 13. So first of all, I have tested positive for COVID, so I'm not coming in this week. I'm feeling pretty fluey, but I've been double vaxxed, so I should be all right. But I'm recording this video lesson for you because we still have lots of work to cover this week, and should be back the week after this week. Should be back next Monday, but we've got 3 lessons to do this week on the nervous system, and that is still going to be in the mock. So I'm recording these video lessons for you, so we can still cover everything we need to. So the first thing is I'd like you to look up in the glossary of your textbook these 4 definitions: resting potential, stimulus, generator potential, and action potential. So look those up in the glossary in the back of your A-level textbook and write them down. Pause the video whilst you do that. Now I also asked you how to think about how they relate to these pictures above. So we can do that now. So resting potential is the potential difference across the neuron whilst the cell is at rest. And resting potential is a little bit like the picture of the dominoes on the left. The dominoes at rest have the potential to fall, and they also have the potential, if they're arranged in this kind of chain, to transmit sort of an impulse from one end of the domino chain to the other. So the stimulus is the thing that causes the change in the environment that then brings about a response. Generator potential, we looked at a little bit last lesson. This is a change in the potential across a receptor membrane. So that might be a pain receptor, a Pacinian corpuscle pressure receptor in the skin, or any other type of sensory receptor cell. The action potential is a brief reversal of the potential across the membrane of a neuron, causing a peak of plus 40 millivolts compared to a resting potential of minus 60. And the action potential is the thing that moves. So the action potential is a bit like this diagram on the right. It is a kind of chain reaction where the kind of resting potential, which was, remember, the dominoes sort of standing upright, is reversed, and it moves along the axon. So well, first of all, before we get into structure and function of neurons, which is our main kind of topic for today, we're going to do a little bit more on what we did, kind of just solidifying what we did last lesson. So here we go. So a nerve impulse is a electrical signal. So the first thing I want to sort of show you is that we can actually measure these. Okay, people haven't made this up. People can actually take tiny little microelectrodes, very, very teeny tiny things, and kind of poke them into axons. And what they do is a bit in this diagram up here. It shows up here, you kind of poke one electrode inside of the axon and one electrode outside of the electron. It's just a voltmeter, really. And it measures the potential difference, not in volts, but in millivolts, so fractions of a volt. And normally, it's about minus 70. I think your textbook actually says minus 60, but I think it varies a little bit, so minus 60, minus 70. So the membrane is polarized with the inside as being negative. In an action potential, it actually goes to positive, so it kind of flips. So first of all, let's recap and look at, again, at how the resting potential is maintained. So we looked last lesson at the sodium-potassium pump, and I found, with Mr. Stones' help, a way of showing you this animation here, which kind of shows you it more clearly. So it's created by the distribution of ions, mainly sodium and potassium. Now, in the membrane of the axon, there are many, many, many sodium-potassium pumps. So sodium-potassium pumps move an unequal number of ions. They move 3 sodiums out of the axon into the extracellular space, but they only move 2 potassiums into the axon. So they put 3 pluses out and only 2 pluses in. So already we're generating a bit of a negative charge on the inside of the axon. But it doesn't stop there. So potassiums have been pumped into the axon. So there's a high concentration of potassiums inside the axon. But there are also channels in the membrane that are specifically open to potassium. Okay, so the membrane itself is kind of permeable to potassium. So potassium ions leak out down their concentration gradient through open ion channels. These green ones here are the ion channels that allow potassium out. So first of all, we've pumped more sodium out and only a few potassium in, 3 versus 2. And then also the potassiums that do come in, they move out down their concentration gradient. So we get even more negative charge building up inside the axon. So that's giving us a lot of polarization. And finally, there are also biological molecules, which we can term anions, such as glucose, proteins, amino acids, that are negative. Okay, so this adds to the negative potential, or polarization, I should say, inside the axon. So all of this generates resting potential, which is polarized and negative, around about 60 to 70 millivolts negative on the inside of the axon. So this is just saying that those potassium ions, they do leave via the channels, even though there is this electrochemical gradient, the fact that there's a polarization negative on the inside of the membrane that you might think would hold the potassiums inside and wouldn't allow them to exit. The concentration gradient kind of force for the potassium ions is larger, so they still exit the axon through open ion channels, causing this polarization. Okay, so the whole thing gives us minus 70 millivolt polarization. Okay, more on that later. So, yeah, your textbook says about minus 60 millivolts in a resting cell. So last lesson we looked a bit about generator potentials, and I wanted to show you this slide. So a generator potential is that small change in polarization caused by a receptor. Okay, and a small stimulus won't cause an action potential. A large stimulus, if it gets to a crucial value, will result in an action potential. And I've added this pink line here. So this small stimulus doesn't get to this pink line. And this one actually also doesn't quite get to the pink line. It's just below this threshold value. But if a big stimulus comes along and it gets above this pink line, then the stimulus is triggered. Okay, because it's an all or nothing response. And if we get to this threshold value, then an action potential will be triggered. So the threshold value is around about minus 50 millivolts. So we can see these failed kind of generator potentials that don't actually trigger an action potential are sort of shown in blue here. But if we get past that threshold, it will go. Now, another thing to sort of talk about in terms of these action potentials is that they're just they're a one size thing. Okay, you don't get big action potentials and small action potentials. You just get no action potential or action potential. So you might think, well, if I'm feeling pain, how does my body know it's like a little bit of pain or like a lot of pain? So how is that encoded in the nervous system? And the answer is by the frequency of stimuli. I'll show you now. Okay, so here's changing the strength of the stimuli. So if we have a sort of medium stimuli, we get an action potential like that. If I was to increase the strength of the stimulus, then we would just get more frequent action potentials, okay? And if I was to decrease the strength of the stimulus, then, well, we might not get any action potentials at all because they don't meet that threshold level. So with a little bit of a stimulus, is that going to be enough? Okay, so medium, we get an action potential, and with a stronger stimulus, we just get more frequent, excuse me, action potentials. Okay, let's come out of that animation. And I'd like you to answer these questions. Now, if you can print this slide, if you have the ability, then you might want to. But if not, let's just call this question 1, answer question 1. Let's call this question 2, answer question 2. And let's call this question 3, answer question 3. Pause the video whilst you do that. Okay, let's quickly review the answers. Basically, this is 3 sodiums out, 2 K plus in, and also K plus channels are open in the membrane, so they move out as well. Over here, basically we have opening ion channels, and they're gated in some way. My favorite are those mechanoreceptors with a little trapdoor. So normally it's sodium ions move in. And then this one, if it's big enough, we call it an action potential. Okay, moving on. So remember we said that common features of sensory receptors is that the cell starts polarized, there are ion channels in the cell surface membrane, they open in response to some sort of stimulus, and ions move in, generally sodium moving in, depolarizing the cell, which causes a generator potential. Okay, so here's another bit more detail on that. Just some more kind of notes for you to write down. Can you copy and complete this text just to fully explain how resting potential is generated using the words on the right now? Pause the video and come back when you've done that. Okay, here they are. Those are the answers. Did you get them right? I think so. And that just kind of sums up what we've looked at in that animation before. Okay, we're now going to move on into some new stuff. All neurons send these action potentials, and we're going to look at action potentials more specifically next lesson. I'll do another separate video on action potentials, but first, let's look at the 3 different types of neurons. There are 3 different types of neurons. You should write these down. Sensory neurons, they carry action potentials from sensory receptors back to the central nervous system. There are motor neurons, which carry action potentials from the central nervous system to effectors, such as muscles or glands. And there are also relay neurons that connect sensory and motor neurons together. Okay, so let's look at each type of neuron in turn. I would like you, if you can print the slide, if not, I'd like you to make the following notes. I would like you to use your textbook, page 48, 49, and draw 3 diagrams of each type of neuron. So you draw the motor neuron, sensory neuron, and relay neuron, and also you're going to make some notes on the function and structure of those types of neurons. Before you do that, though, you should do a little mind map on neurons in general. So what are the general common features? For example, they have a lot of mitochondria because they're very metabolically active. They all have a cell body, that kind of stuff, okay? So page 48, structure of neurons, is what you're going to do, get the kind of general features. Then page 48, the diagrams of neurons. And page 49, the differences between the types of neurons. Okay, that should probably take you about 10 minutes or so, maybe even up to 15, depending on how detailed your drawing is. So pause there and come back when you're finished. We were going to do this in class as well, making a model of one in plasticine, but probably don't do that now. Here we go. General features. They are long cells, many gated ion channels, lots of sodium-potassium pumps. They are polarized. They have a cell body with lots of nucleus, mitochondria, ribosomes. They have dendrites, which connect to other neurons, and they have an axon, which carries the impulse away from the cell body. So dendrites carry the impulse towards the cell body, axon away from the cell body. Some neurons also have a fatty layer, which is called myelin, which insulates the axon, and it helps speed up the message. More on that later. Here are some diagrams which you should have drawn, and the main features that you should have written down. Okay, so check that. You can green pen it, and then we're going to move on. We're going to go 2 more tasks to do. So the next thing. Myelinated versus non-myelinated. If you can, draw a diagram a bit like this. So a myelinated neuron has this kind of, I don't know, almost like sausage roll appearance, with the sausage being the axon in the middle and like this pastry wrapped around it to kind of insulate it. It's not pastry, obviously, it's a Schwann cell. And that Schwann cell has lots of myelin. That is a fatty material that insulates and kind of protects the electrical message within the axon, okay? So, draw a diagram, put the advantages of myelination and the disadvantages, make a note on how the action potential travels and where do you find them. So here's the table here, which you can kind of make all these notes. Advantages, disadvantages, how does the action potential travel in the 2 different types of neuron, and where would you find them? Okay, so pause the video if you haven't done that. And we're moving on to our final task. Right. Final task, do these 3 pages that I'm setting you on Teams. Hopefully you can print them if you can. If not, you can just kind of write the answers down So questions 1 to 4 on page 26, and then so on and so on. But it'd be great if you could print these out. I'm attaching them as a PDF to your assignment. And once you've done that, I will also upload the answers to that as a separate thing, but I think I'll leave you waiting for that. I'll upload the answers in your second lesson at the beginning so you can mark it there. So consider this your sort of mini homework. And then the second lesson of the week, which will also be video because I'm out for a week, is going to be on the action potential, okay? So all the best, and I'll see you when I get back. Bye-bye.